Welcome back. Let's talk a little bit about cell membranes, and in particular I want to focus in this discussion on the structure of cell membranes. What are the components? And we'll also talk a little bit about, we'll introduce the ideas associated with some of the functions of membranes as well. First of all, when we talk about this membrane, you want to think about it as forming a barrier between the extracellular environment and the intracellular environment. And so we can think of membranes as a way in which the cell helps to organize the chemical activities of the cell, keep the chemical environment inside the cell, right, the aqueous solution there, distinct from the aqueous solution surrounding the cell. We could think of membranes in general as the way in which cells are going to organize their metabolic activity. Remember, when we're talking about these eukaryotic cells, we're going to find numerous membranes throughout the cell. And each of these membranes is going to have basically the same structure. We're going to focus in this discussion on the cell membrane, but you want to apply this to the membranes of the ER or the Golgi or the nuclear envelope, and they're all going to have similar construction, and they're going to have some similarity in function as well. Now, one of the things you want to make sure you also realize is that when we talk about prokaryotic cells, remember, they only have a cell membrane. So as you think back to the different functions of organelles found within the cell, you want to think about how the prokaryotic cell has to use their cell membrane, their cytoplasmic membrane, to carry out virtually all the functions of the organelles of a eukaryotic cell. Now, membranes, one of the key features is we say that they are a selectively permeable membrane, a selectively permeable barrier. Remember, what that means is that some things can get through and some things can't. In a very simple way, you can think of selectively permeable barriers as being things like the coffee filter that you use at home, or the window screen in your window. Some things can pass through, some things can't. This selectivity of the membrane is going to allow the cell to control the movement of substances into and out of the cell, from extracellular to intracellular, from intracellular to extracellular. Key control function of the cell. Also, we're going to find that membranes will hold groups of molecules in them, for example, like enzymes, that might do some type of related function. So there's going to be a number of cases. We're going to talk about membranes having specialized groups of molecules embedded in the membrane, carrying out some specific specialized component of the metabolic activity of the cell. Now, what's the primary constituent of the membrane? It's going to be a phospholipid molecule, and we're going to find these phospholipids in what's referred to as a bilayer, 2 layers of phospholipids. Let's review quickly the structure of phospholipids. Remember, they had that head region where the phosphate group and the glycerol portion of this molecule is found, and we expect that portion of the molecule to be polar, hydrophilic. If you didn't know that ahead of time, you could come to that conclusion because of the concentration of oxygen in that region. Remember those electron hogs? They tend to create what? Polar covalent bonds, and therefore regions with charges. As a result, we'd expect that part of the molecule, the polar portion of the phospholipid, to be hydrophilic, able to mix with water. Then we have the 2 long hydrocarbon chains of the fatty acid, and we tend to think of them as being highly nonpolar, right? Take a look at the illustration. You can see no oxygen there, no nitrogen there. I wouldn't expect those bonds to be polar covalent bonds. I wouldn't expect there to be charges associated with this fatty acid tail region of the phospholipid, and therefore I'd expect that part of the molecule to be hydrophobic, right? So here we have a good example, one of the best examples, you might argue, of an amphipathic molecule. Remember, that term means a molecule that's both hydrophilic and hydrophobic. Or we could say it another way and say, here we have a molecule that's both polar and nonpolar, an amphipathic molecule. Well, look what happens when I take phospholipids and I place them into an aqueous environment. The phospholipids automatically arrange themselves so that their hydrophobic tails are pointing towards each other, and the hydrophilic heads are pointing toward the water environment. So the heads face outward and the tails face inward. If we think of this sandwich, the bilayer of the phospholipids. Remember, the heads are the hydrophilic component, the tails the hydrophobic. Now here I've inserted an illustration to remind you that We don't want to think of this structure as just being associated with eukaryotic cells like your own, but also we want to realize that this is the same type of structural arrangement we'd expect to find in prokaryotic cells. And so you can see that once again we're showing that the membrane of a prokaryotic cell is also one of these phospholipid bilayers. And as you can see from one portion of this illustration down here, that there's a distinctive way of representing those phospholipids. Notice that really isn't very different than this representation. You're going to see phospholipids drawn in a variety of ways. Here's another representation. But notice that all of them have the basic feature of showing the hydrophilic head and the hydrophobic tail regions. All right, so one of the things that you might find as you cover more topics in biology is that people will talk to you more about the archaea. Remember the archaea, another type of prokaryotic cell. And one interesting feature about the archaea is that they also have a cell membrane, as you might expect, but instead of using phospholipids, they use a different family of lipids to make up their lipid bilayer. Notice, their membrane is still a lipid bilayer. It's still made out of amphipathic molecules, but now they use a molecule called a glycerol diether. I won't ask you to learn or memorize the structure of that, but I did want you to realize that there are alternatives to using phospholipids for making membranes. That alternative shows up in the archaeal group. And it's a strange molecule compared to these phospholipids, but one thing you might expect them to be is some type of lipid, and sure enough, these glycerol diethers are lipids, and the membrane of the archaea is a bilayer of those molecules. Interesting how basically the same kind of structure is found in those two groups of prokaryotes, but with a distinctive, individualized molecule found in each. Now, bacteria, eukaryotic cells, we use the phospholipid bilayer, and so we're going to talk more about that. Now, this membrane, this phospholipid bilayer, we want to think of it as a fluid mosaic. Let's take both of those words and talk about them a little bit. First of all, the fluid part. I want you to realize that these membranes are flexible. They're not rigid, hard structures. You don't want to think of something like a hard plastic surface on a ball. Instead, you want to think of something that, you might think of as being as flexible as a drop of oil. It can change shape, it can bend, it can flex, it can stretch, it can form depressions. So we're talking about a structure that's quite fluid in that respect. Now also, the fluidity is something that you can think of as being the composition of the membrane. By this I mean that it's not the same membrane from one time period to the next. Your cell membranes are constantly having molecules added to them and molecules taken out of the membrane. So I mean fluid in the sense of its composition changing over time. So two interesting qualities of fluidity associated with this membrane. Let's focus now on the mosaic nature of the membrane. Now, a mosaic is a picture made up of lots of individual little parts, and we're going to find that the membrane has more than just the phospholipids present. As you can see here, I'm suggesting to you that the phospholipid bilayer has embedded in it other lipids, in particular certain types of steroids, like cholesterol. By the way, if we were looking at prokaryotic cells, we might see an additional family of lipids called hopanoids. So there can be steroids or these interesting hopanoid molecules added to the phospholipid bilayer. In addition to that, we expect to find proteins embedded in the membrane as well, or stuck to the surface of the membrane. Attached to some of the components of the membrane, there might be sugar chains. So we might have lipid molecules with sugar chains attached, glycolipids. We might have protein molecules that are part of the membrane with sugar chains attached. We'd call those glycoproteins. Interestingly, those glycoproteins and glycolipids have as a primary function cell recognition, and that might be a foreign idea to you right now, but let me just give you one quick example. If you ask me what my blood type is, I might say to you, I'm blood type AB. What I'm really describing to you are the kinds of sugar chains that extend from my red blood cells. Type A is one particular version of a sugar chain, a type of glycolipid molecule. Type B, a different sugar chain, another kind of glycolipid molecule. And as you probably know, you can't receive any kind of blood in transfusions. Your body actually can recognize the difference between your cells and some foreign cells. And so when we talk about cell recognition, that's the way you want to think of it, is that we're talking about the way in which the immune system of our body can recognize which cells are part of us and which cells are foreign material. And in many, many cases, not all cases, but in many cases, that's determined by the glycolipids and glycoproteins sticking out of the surface of a cell. So cell recognition is that idea. An immune system able to recognize or determine where a particular cell is from. Now, the proteins have an enormous number of functions. And so I'd like to take a moment just to talk a little bit about the different ways in which we designate some of these proteins. Now, remember, what we're saying is we have a phospholipid bilayer. Okay, so there's my two phospholipids making up the bilayer. I might find protein molecules that extend the whole way through that membrane. Since the protein's embedded in the phospholipids, we would call it an integral protein. Since it passes the whole way through both layers, we call it a transmembrane integral protein. Some integral proteins don't pass the whole way through, though. They're simply embedded in the phospholipids. So we're going to see these types of proteins associated with the phospholipid bilayer. They're going to be integral, which means they're stuck into the phospholipid bilayer, either passing the whole way through, transmembrane, or part way through, embedded proteins. There's also going to be proteins that are just going to sort of lay on the surface of the membrane. They might be attached here. They might even lay on the surface of other proteins. Usually these proteins are weakly attached with electrostatic attractions, hydrogen bonding, maybe ionic bonding. They're called peripheral proteins. Peripheral proteins. And then there's another family of proteins, and they actually have a portion that is lipid, and then a portion that is protein. So we would refer to those as being lipid-anchored proteins. So notice the proteins come in a variety of different forms. At least we could think of them as being defined by their position, maybe, their location. And in one case, with the lipid-anchored ones, in terms of their composition. And we're going to find that the proteins in membranes are going to differ in different membranes. And that's what's going to give the different portions of a membrane, or the membranes of different organelles, different functions. The kinds of proteins that are associated with the membrane. All right, so please pay special attention to membrane proteins as you're thinking about membrane structure. Oh, there'll be lots of examples we'll come across as we go through other material in this semester. There's another representation of the membrane, a little bit nicer than the one I just drew. Notice a few things, if you will. We have some nice examples of integral proteins and peripheral proteins. Think back to the illustration I just had. Think about the definition I gave for them. So, integral, embedded, peripheral, on the surface. Notice also, we have some nice examples of glycoproteins, right? A sugar chain attached to a protein. We have glycolipids, a lipid molecule with a sugar chain attached to it. We also have, notice, there's my steroid, cholesterol. The cholesterol is very important, by the way. In many cases, all we ever hear about cholesterol is when somebody has too much or an excess of cholesterol. But you want to realize it's a key component of cell membrane structure. The cholesterol actually helps to maintain membrane flexibility at lower temperatures. And interestingly enough, it helps to hold membranes together at slightly higher temperatures. So the cholesterol is there as a mechanism for our membranes to maintain their appropriate function at lower and higher temperatures. So you can see that we have a number of different components of the membrane represented. And of course, the key component are these multiple phospholipids making up the bilayer. Notice, however, that they also show in this illustration that on the cytoplasmic side of the membrane, components of the membrane may be attached to the cytoskeleton. And on the outside of the membrane, components of the membrane may be attached to the extracellular matrix. So now don't think of this membrane as just floating around in pure water. Instead, think of it as interacting with both the extracellular environment and the intracellular environment. Okay? Fluid, mosaic. Make sure you practice thinking about how you would describe those two features of membranes. Now, the proteins I mentioned are going to have a variety of functions, and one of the important functions they have is as cell junctions, holding cells together. You may remember that I talked about things like the attachment points between cells, tight junctions, gap junctions, desmosomes that help to hold cells together. Those were protein in nature. Also, we expect to find that many of the transport processes through cell membranes are going to be regulated or controlled by proteins embedded in the membrane. So transport, another important function of membrane-associated proteins. Now, what we're going to also find is that in many cases, the proteins that are part of a membrane may work as an enzyme. So we might have proteins embedded in the membrane carrying out enzymatic function. And some proteins are going to work as receptor molecules, molecules that can be attached to by something from the outside environment. In this particular illustration, it's called a messenger molecule. You might also see the general term used for any molecule that binds to a receptor, a ligand. And these receptor molecules, as their name implies, receive things, but the key feature of them is that they then cause some kind of change in the internal cellular environment. The binding of that ligand, the binding of the messenger, will cause what is sometimes referred to as signal transduction. A message from the outside causes a chemical change on the inside of the cell. Many of the hormones that you might be familiar with have these kinds of actions. The neurotransmitters sent from one cell to another cell in nerves have this kind of function. So another important functional role for proteins. So make sure you review those categories of protein function I've suggested for membrane proteins: attachment, transport, enzymatic function, receptor function. 4 key categories of protein function associated with membranes. All right, so let's back up and take a look at where we've been. We said that we were talking about primarily a phospholipid bilayer, and I also indicated to you that even if membranes aren't composed of phospholipids, that membranes will be composed of a lipid bilayer. Right? Remember what I mentioned about the Archaea cells? They use a different kind of molecule, but it still is an amphipathic lipid. We said that the cell membrane is a fluid mosaic, composed of multiple subunits, different kinds of parts. Fluid in the sense that it's constantly changing and it has a high degree of flexibility, stretchability. We said that 2 of the key things that are included in the membrane along with the phospholipids are other lipids like steroids, or in some bacteria, another class of lipid called hopanoids, and proteins. And remember, we noted that proteins have a lot of specialized functions when they're in a membrane. Finally, we said that cell recognition molecules might extend from the surface of membranes in the form of glycolipids and glycoproteins. All right, spend some time reviewing this. Look for the function of membranes in a variety of the topics we talk about in the future. Go back and review some of the organelles we discussed in the past. Thanks very much. Look forward to talking to you next time.